Okay, before discussing the epithelial tissue, I saw that there are some questions on Zoom on the live chat, and Hani already answered some of those questions. She will help me a lot by answering questions from the live chat, and she talked during this break to me and about organizing already the Canvas conferences, the PA review sessions. But now I have a big task to go to the epithelial tissue lecture in a reasonable time. I'll try to do that. So there are these four basic tissues, the epithelial tissue, the connective tissue, muscle tissue, and the nerve tissue. And the order in which these tissues are discussed is primarily decided by how histology was taught in the past. It's usually this order: the epithelial tissue, connective tissue, which does not cover all the forms of connective tissue. We'll discuss that. We'll discuss that next Thursday. But then muscle tissue and then nerve tissue. And after that, we begin with studying the systems, the nervous system, the integumentary system, and others. So at the end of this lecture, we should be able to understand what epithelia are, how their polarized structure relates to their function. So epithelial cells have a polarized structure. The interior of the cells, it's polarized in a certain way. And not only that, they have domains of the plasma membrane that consist of different components. So even there is a polarity of the plasma membrane. We will look at the different types of epithelia and their location. We will learn to recognize the different types of epithelia and histological preparation. And that is actually an important task that we need to achieve today. And after you study this lecture in the coming weeks. We will look at the two types of glands that are actually formed by epithelial tissue, and we'll discuss the exocrine glands and the endocrine glands. We will look at the differences in the organization, nature of secretion, mode of secretion between the different types of glands. We will understand the cell polarity, the role of the basement membrane. The basement membrane, it's a structure, an extracellular structure, and we'll see that it is made of constituents that are produced by other cells, connective tissue cells, but it also incorporates proteins that are produced by the epithelial cells themselves. We will talk about the different types of intercellular junctions. The epithelial cells are associated with each other quite tightly, and there are different types of junctions, and most of them are there to the attachment junctions. They keep the cells attached to each other, but then there are the tight junctions that seal actually the membranes between these cells, and the gap junctions that form connections between the cytoplasm of the cells that are associated by gap junctions. We will then look at the structure and function of specializations at the apical domain, the microvilli that are made of actin filaments, the cilia that are made of microtubules, and the stereocilia that are not real cilia in terms of what cytoskeletal elements they contain. They contain as cytoskeletal elements the actin filaments. And then I'll mention a few things about the epithelial regeneration and the pathology. So what is the epithelial tissue? It is composed of closely aggregated polyhedral cells adhering strongly to one another and to a thin layer of extracellular matrix. It forms cellular sheets that line the cavities of organs and cover the body surface. And if you culture epithelial cells and they reach the confluency, you may at some point remove the cells in the culture as sheets of cells because they are tightly attached to each other. The epithelia line all external and internal surfaces of the body. So we are covered both on the outside and on the inside by the epithelial cells. And this makes it this makes us reach the conclusion that nothing really can enter our body until they pass through the epithelia. We swallow something and that may be eliminated almost untouched without really penetrating into our organisms, because in order to do that, they need to pass through the epithelial lining of the digestive tract and enter in our organisms. So the principal functions of epithelial tissues include covering, lining, and protecting surfaces. And one example is that of the epidermis, the outer part of the integument. Absorption, like the intestinal lining, and secretion, the parenchymal cells of the glands. This is a slide I will try to go as fast as I can through it because it describes the different types of epithelia. And what you see in each of these images are the epithelial cells that lie actually on the basement membrane. This reddish line that you see here in each of these drawings is the basement membrane. And in the all epithelia that are simple epithelia, the cells, all the cells are attached to a basement membrane. In the stratified epithelia, the basal layer is attached to the basement membrane. So the epithelia are classified according to the cell shape and the number of layers. In terms of number of layers, I mentioned already that they can be simple epithelia made of one single layer of cells or stratified epithelia. Usually it's a double epithelium that is made of two layers of cells or more layers of cells, as it is in the stratified squamous epithelium and in the transitional epithelium. According to cell shape, there is the simple squamous epithelium, cuboidal and columnar epithelium. And you can realize what this is the nomenclature. The squamous epithelium is the cells appear like squames. They are very, very flat. And the nucleus often bulges out of the plane of the cell. The cuboidal epithelia have a cuboidal shape, and the columnar epithelia have a columnar shape. Then the stratified epithelia, the cuboidal and the columnar are usually double epithelial layer with only the basal layer resting on the basement membrane. The stratified squamous epithelium has several layers, and these layers are produced from cells that all originate in the cells in the basal layer. The basal layer contains the cells that divide, and as they divide, they reproduce each other and replace the cell in the basal layer, but the other daughter cell, it's pushed upwards and advances towards the surface of this stratified squamous epithelium. And this one can be of two types. In one type, the cells are viable till the top, and their nuclei and the entire cell structure can be easily distinguished. And it's the case of the non-keratinized stratified squamous epithelium. However, the epidermis, it's a type of epithelium, of stratified squamous epithelium, that is keratinized. And the top layers of cells actually die, and the keratin, which is the intermediate filament in epithelial cells, aggregates into bundles, tightly associated bundles that are meant to protect the epithelial, the cells that lie underneath this keratin layer. Then there is the pseudostratified epithelium that is, it appears according to the position of the nuclei in this epithelium. It looks like the cells are organized into layers of cells, but there is really one single layer of cells, that all cells touch the basement membrane, but not all cells reach the surface. So this is the pseudostratified epithelium. And then there is the transitional epithelium, which is a stratified epithelium that changes the appearance dependent on the degree of stretching of the epithelium. It's also called urothelium because it's present only along the urinary tract, in some parts of the urinary tract. And then, if you imagine that it is part of the lining of the urinary bladder, when the urinary bladder it's empty, then the epithelium appears compacted. When it fills in, the cells become stretched, and they change also the shape from being sort of like dome-like cells, cuboidal cells. They may become even squamous cells in the filled bladder. Now let's take a look at each of these, say a few words about each of these epithelia. The simple squamous has functions in diffusion, filtration, and gas exchange. And it looks as shown here in these images. This is the mesothelium. It is here, this is the mesothelium in the epicardium. The epicardium is the outermost layer of the heart, and that is lined by an epithelium that is simple squamous. You see that the nuclei are elongated, and the cells are really have no height almost. Locations in the kidney renal corpuscle, lung alveoli, the endothelium that lines the interior of the heart, the endocardium, and the intima of the blood vessels is also a simple squamous epithelium. And the mesothelium lines actually the body cavities in the pericardium, the peritoneum, that is the cavity that is present in the abdominal cavity, and the pleura that surrounds the lungs. The pericardium is actually the sac-like structures that surrounds the heart. And here I have to stop and tell you that we're at the beginning of the course. There are many things that I cannot go into details right now. I'm mentioning them, but I will come back as we go along with the various lectures. These are other examples of simple squamous epithelia. You see this is in the renal medulla, in the medulla of the kidney. And you see that the cells have actually almost no height. This is actually the thickness of the cell. You only see the nuclei bulging out of the plane of the cell. This is another image from the kidney. In this case, it's the kidney cortex, where the Bowman's capsule, it's lined by a mesothelium, by a simple squamous epithelium, and it is this one. This is the mesothelium that is part of the serosa in the urinary bladder. So the inner part of the bladder is lined by the transitional epithelium, the outer part by the mesothelium. I mentioned also that the simple squamous epithelium is a cell from which cancers often originates, and the severe cancer, the mesothelioma, originates in the mesothelium that covers the pleura surrounding the lungs, and it is a cancer that it's triggered by the presence of asbestos particles that are inhaled. In the past, especially when asbestos was largely used in building the houses. And this is a severe cancer, a severe form, deadly form of cancer, because it metastasizes. The simple cuboidal epithelium has functions in secretion and absorption, locations, the small glandular ducts, kidney tubules, and the thyroid gland. The follicular cells in the thyroid gland that you see here are made of a simple cuboidal epithelium. And so this would be the thyroid follicle. You see all these cells. Here, I have to tell you, you can't distinguish really the cells. Where are the boundaries of the cells? But you see the nuclei, and you imagine that this is one cell, this is another cell, and you get the feeling that these have actually a cuboidal shape. This is a simple square, cuboidal epithelium, although in these regions you see two layers of cells. Here you also see two layers of cells. But this is because these epithelial cells line the follicles, and the follicles are tightly packed with each other. And here the appearance as a dual epithelium is caused by two follicles being tightly associated with each other. This is another example. In the kidney, these are the convolutive tubules, proximal convolutive tubules in the kidney. Here again, you have to imagine this is one cell, this is another cell, this is another cell. I told you that you have sometimes to use your imagination to interpret the images that you see in histology. There is also a pathology of the simple cuboidal epithelium, and the pathology it's highly represented in the thyroid gland, and it is metaplasia. Metaplasia is actually a change in the shape of the epithelium. That is actually a functional adaptation. If this thyroid glands, if they secrete large amounts of thyroid hormones, then the cells become larger and they become calander. If they, in cases of hypothyroidism, the cells, they undergo metaplasia into squamous cells. But this is a functional adaptation that it's not malignant and it is reversible. So this would be the drawing of such a type of epithelium. The simple columnar epithelium, what you see here, it's the lining of the jejunum, which is the middle section of the small intestine along the digestive tract, and you see that the cells are columnar in shape. This is the one surface, and you can see that there is something staining here lightly at it appears tightly attached to these cells. That is the brush border made of microvilli, and we'll see what the microvilli do. We'll talk. I'll tell you a few things at the end of this lecture. Functions in absorption and secretion, locations in large ducts, digest and along the digestive tract. And these cells that you see here are actually the goblet cells. The goblet cells don't stain with hematoxylin and eosin, but here they have a bluish stain. And this is because here they also secrete a negatively charged mucus that contains sialic acid that is negatively charged. And this is why they are likely basophilic. Then the stratified cuboidal and columnar epithelia, they contain two layers. Here again, how do you know that these structures, which are this is a cross section of a sweat gland duct, contain two layers of epithelia? You see actually two concentric layers of nuclei. So this is clearly a structure that it's made of a double layer of epithelium. Locations in stratified cuboidal in sweat gland duct, mammary gland ducts, stratified columnar, lobar ducts of large salivary glands, male urethra, the pharynx, the conjunctiva inside of the lids. And this is, for example, the palpebral conjunctiva inside of the eyelids. And this is actually a very, very delicate epithelial lining. This is a stratified columnar epithelium in the male urethra. This is a mucosal gland in the esophagus. And this structure actually is a duct, a quite large duct associated with that mucosal gland. And you see two concentric layers of nuclei. This is a sweat gland. The stratified squamous epithelium, I mentioned already that it contains several layers of epithelial cells, and all layers in the upper all cells in the upper layers are generated through division of the cells in the basal layer. And this, as they are generated, they are moving in the upper layers. And they, at some point, they reach the surface. But the cells at the surface, they desquamate. They have a relatively short half-life, and they will be removed from the surface of the epithelium and replaced by the cells that come from the inner layers. I mentioned already that some stratified squamous epithelia are keratinized, as there is the stratified squamous epithelium in the epidermis, and there the keratin layer is also desquamated, and the cells in these upper layers are dead cells, but they are mechanically very resistant, and we'll talk more about this type of epithelium I think 3 weeks from now when we discuss the integument. Then the non-keratinized one, the cells in the upper layers are still alive. And I have to mention, however, that if you would look at these lower interior layers in the stratified squamous epithelium, you will see that the cells are not squamous. They are cuboidal, almost columnar. But the name of such stratified epithelia comes from the shape of the cells in the uppermost layer, and they are really squamous cells. The pseudostratified epithelium. It's made of a single layer of epithelial cells that appears multi-layered. And this is an example. I think that this is from the virtual microscope. It's the epithelium lining the trachea and most of the respiratory tract, which is a ciliated columnar pseudostratified epithelium with goblet cells, and these are the goblet cells. These epithelia are ciliated, and you see the cilia at the surface. And the nuclei are at various levels here, but they all cells actually touch the basement membrane that it's present there. Functions in absorption and secretion present in large ducts, respiratory tract, and male reproductive system, the epidermis and the vas deferens. And this is another image that shows you the respiratory epithelium. All these cells actually touch the basement membrane, but some, the basal cells, don't reach the upper part. Then the transitional epithelium of urothelium. It's urothelium because it is present only in the urinary tract, on some portions of the urinary tract. It has functions in protection and distensibility. And this is the unstretched epithelium, transitional epithelium. This is the stretched one. You see that the cells have been stretched so much that they appear almost squamous cells. To keep in mind for the exam is that the transitional epithelium has the same number of layers of cells in both the distended and the stretched and the unstretched state. So the distended urothelium appears only to have fewer layers with flat squamous-like apical cells. These are histological preparations that show, you see that the cells in the uppermost layer, they are quite big cells. They are called umbrella cells or dome cells, and we'll talk a lot more about them when we discuss the urinary tract. Now about the glandular epithelium. The glands, they develop from an epithelium, and there are two types of glands, the endocrine glands and the exocrine glands. You have to imagine, let's say that this is a stratified epithelium, and during embryonic development and fetal development, cells in a region of this epithelium start dividing, and they multiply, and they sort of penetrate outside of the epithelial lining and generate, and at some point, a cavity. That cavity becomes the lumen of the secretory unit that remains connected to a duct. And in this way, the exocrine glands are capable to produce a secretion that is delivered to the site where the secretion needs to reach, through a duct. The endocrine glands do not have glands. The endocrine glands produce hormones. The initial stages of formation of the endocrine glands is the same as for the exocrine glands. But at some point, this portion of almost of tightly packed cells completely detaches from the epithelial lining from where they came from. And they either develop a cavity and generate a follicle and are lined by follicular cells, or remained as a packed structure of cells. And begin synthesizing the hormones and secreting the hormone. The hormone, it's not secreted in a follicle inside the lumen. It's secreted at the basal region of the cells in the extracellular space, and the hormone immediately enters capillaries. The endocrine glands are surrounded by capillaries, and the hormone is distributed to the sites that they need to reach through the circulation, through blood. So the exocrine glands contain ducts. The endocrine glands do not contain ducts but are very vascularized. Examples of endocrine glands are the thyroid gland. The pancreas contains the islets of Langerhans. So what you see here are the mass of exocrine glands that make the bulk of the pancreas, but here and there are the islets of Langerhans that contain the endocrine cells that secrete hormones related to digestion, like the glucagon, insulin, somatostatin. We'll talk more about the pancreas when we discuss the digestive tract. The exocrine glands, they don't have ducts. The secretion enters immediately into the blood vessels and is distributed throughout the body through blood. Salivary glands, I think that I now what I told you refers to the endocrine glands. The exocrine glands, they have ducts. The secretion is produced into a lumen that is at the center of a secretory unit, and the secretion is then pushed into ducts and delivered through ducts to the site that they have to reach. And examples are the salivary glands and also the pancreas. The pancreas is a mixed gland. It contains an exocrine part and an endocrine part. Now, the secretory units and the ducts form what's called the parenchyma of the gland, of the exocrine gland, and they are embedded in the connective tissue that forms the stroma, that supports actually the secretory units and the ducts. Now, this is a very important slide because it discusses the classification of the glandular epithelium. I will go very fast through it, but I'll try to be very precise. According to the timing of secretion, the secretion can be constitutive or regulated. Constitutive secretion is the one that immediately it's secreted from the cells as after it is produced. The regulated secretion, the secretory product, accumulates into vesicles, and those vesicles fuse with the plasma membrane and release the secretion only when a factor comes, a signal comes and triggers the secretion. According to distance traveled by the secretion, there is autocrine secretion that acts on the cell that has produced the secretion as it is in platelets. Paracrine secretion, where the secretion acts in the surrounding. The endocrine secretion that is produced by the endocrine glands that have no ducts and produce hormones that are delivered through circulation, and the exocrine secretion that is carried by ducts to the sites where the secretion needs to get. According to the nature of the secretion, there is a serosecretion. The serosecretion is a protein serosecretion, proteins that are not glycosylated. It's a watery secretion. The zymogen granules in the pancreas that we discussed already, they contain a serous secretion. The zymogen is a serous secretion. The mucose secretion, it's rich in glycoproteins. the goblet cells produce and secrete a mucus that is proteins heavily glycosylated, sugar-rich secretion that is stainable with periodic acid sheath stain. There is mixed secretion by mixed acini. The mixed acini contain some cells that are serous cells and produce a serous secretion, and others that are mucous cells. The serous cells are usually distributed at the periphery of the secretory units. And then there is the hormonal secretion that I've discussed already. Another important thing is the mode of secretion. The merocrine secretion is the most common one. It's typical exocytosis, when a vesicle filled with the secretory product comes next to the plasma membrane. The membrane of the vesicle fuses with the plasma membrane and releases the secretion into the extracellular space. The holocrine secretion is the one where the entire cell disintegrates and in this way releases the secretory product. And there is also an intermediate form of secretion, which is the apocrine secretion, where chunks of the apical cytoplasm detach and are released. Chunks of the cytoplasm, not the entire cell, it's ruptured. And it's really important to know also for the exam. So the fusion of the synaptic vesicles containing the neurotransmitter with the plasma membrane at the synaptic terminal, it's typical exocytosis and it is merocrine secretion. I'll come back here with regard to the cell number. There are unicellular and multicellular glands. The unicellular, the prototype is the goblet cell. It's just one cell that releases the secretion. Multicellular are the pancreatic acini, where the cells all secrete their product into the lumen that enters then into a duct. And according to the shape of the secretory unit, they can be tubular, acinar, and alveolar glands. They can be simple or compound glands based on the ducts that they possess. They may be simple, where there is one single duct that carries the secretion to the destination, or compound, where the ducts branch. Now, these are examples of serous glands. The serous glands, they stain eosinophilic. This is convex of in pancreas. The cytoplasm contains the zymogen granules filled with the zymogen. That is a protein, a watery secretion. These are examples from a salivary gland where there are serous acini, there are mucous acini. Some of them are mostly and almost entirely mucous acini. And the cells, the mucous cells, they don't stain well with H&E because they contain a sugary secretory product. And then there are the mixed acini where the serous cells are organized in the so-called serous demilunes, half moons at the periphery of the acini. So here the secretion is a merocrine secretion. Now, this is also an important slide. It's about the types of secretion in the mammary gland. The mammary gland is a compound tubulo-alveolar gland, and it secretes what? What's the secretion of the mammary gland? It's the milk. What does the milk contain? It contains protein it contains sugar, which is lactose, and it contains lipid. The protein and the sugar, they are secreted through merocrine secretion, typical exocytosis. The lipid is secreted through apocrine secretion. Chunks of the apical cytoplasm are secreted into the membrane of the secretory units. Now, this slide tells you a few characteristics of the epithelial cells, and I have to apologize. This is always happening. The introductory class meeting lasts a bit longer. And I had to tell you also about the structure of the course, so I still keep you a few more minutes to finish the lecture. So characteristics of the epithelial cells: high cell extracellular matrix ratio. The cells, at least in the basal layer, rest on a basement membrane. Cells have polarity. They attach tightly to each other. Epithelial tissue is avascular. The blood vessels do not penetrate within the epithelium. They are metaplastic, and you know what metaplasia means. They are change reversibly their shape. They regenerate very, very easily, derived from all three germ layers. And when this is the trilaminar disc that forms during the gastrulation in the developing embryo. It contains an ectoderm, a mesoderm, and an endoderm. Epithelia originate in all these three layers. And importantly, the intermediate filaments in the epithelial cells are the cytokeratins. And I mentioned already the keratin that develops in the keratinized stratified squamous epithelium comes from the cytokeratin filaments that are present in this epithelium. Now, about the basement membrane, I mentioned already that the cells, the epithelial cells, rest on a basement membrane, and they are quite tightly associated with this basement membrane that is an extracellular structure. And the basement membrane contains many glycoproteins. This is why in this image it's stained very nicely with the PAS stain. And these are the convoluted tubules. These are the cells that form them, that mostly are the proximal convoluted tubules. But the PAS stain primarily stains the basement membrane on which these cuboidal cells are rest. The basement membrane serves as a selective filtration barrier in the kidney, and this is the renal corpuscle. What's in the renal corpuscle is the glomerulus, and the glomerulus is the site of filtration of the blood, and into a filtrate that later on becomes urine. But the filtration barrier is made by the basement membrane. It is important to know the layers that form this basement membrane. The basement membrane, that is the part of the basement membrane that is close to the epithelial cell, it's the basal lamina. And the basal lamina in transmission electron microscopy has a lighter layer, the lamina lucida, and a darker layer, the lamina densa, but both are parts of the basal lamina. Then there is another outer layer of the basement membrane that is called reticular lamina. It is still part of the basement membrane. But what the main difference is between the basal lamina and the reticular lamina is that the components of the basal lamina are synthesized and released from the epithelial cells, while the components in the reticular lamina are synthesized and released from cells of the connective tissue, essentially the fibroblasts. And I draw your attention to the fact that the connective tissue contains a lot of collagen and many types of collagen, filamentous forms of collagen that are secreted by the fibroblasts. But the basal lamina contains a non-filamentous type of collagen, the type 4 collagen, that is actually produced and released by the epithelial cell. Now, I mentioned the epithelial cell polarity. This is a nice drawing that shows a cell that rests with the basal part of the basement membrane. It has lateral portions of the membrane, and these lateral portions are actually associated with next to the lateral membranes of neighboring cells. Then there is this apical region, and the apical region, it contains those microvilli that are projections of the apical plasma membrane, and they increase the absorptive surface there. But the polarity is visible not only inside the cell, where the nucleus is usually at the basal part of the cell, the endoplasmic reticulum and the Golgi then follow, and towards the apical region are the secretory vesicles. If this is a pancreatic cell, this would be the secretory vesicles, the zymogen vesicles, or the zymogen granules. But what's most importantly is that the cells are separated from each other, but they attach sometimes very tightly to the junctional complexes. The junctional complexes are towards the apical region, and the cells are attached that tight to the tight junctions that the components that are of the membranes in the apical part of the membrane cannot pass through the tight junctions and reach the basolateral membranes, and vice versa. Now let's very fast go through these three types of junctions, and I begin with the attachment junctions. The attachment junctions, they can be of two types. They can run and like a bell throughout the cell and connect this cell, attach this cell to the neighboring, to all the neighboring cells, to all parts actually of the membrane. And those are the belt desmosomes or zonula adherents. And you see, as the drawing shows, that there are structures that are formed from cadherins and catenins that attach the cells strongly to each other, but there is still space in between the cells. There are these spot desmosomes or macula adherents that are they form in spots. They are present here and there along the membrane of the epithelial cell. But they also attach the cells strongly to each other, but there is still a separation between the plasma membranes of these cells. The belt desmosomes attach to actin filaments, the spot desmosomes attach to intermediate filaments. And they all contain the cadherins and the catenins as major protein components that maintain the interaction between the cells. Then there are the tight junctions or zonula occludens, and they run also like a belt. It's actually like a network of structures that tightly attach the membranes of the neighboring cells in such a way that the membranes appear almost nailed to each other. They cannot, nothing can pass from the apical region to the basolateral region through the tight junctions. Main components of the tight junctions are the occludings, the clothings, and the zonula occludings. Those are 0, 1, and 0, and there are also at least two types of occludings. Occluding 1, the first to be discovered, and occluding 2, that was discovered more recently, still quite some time ago, that and the person, the researchers who discovered that occluding 2 is here in this room right now. It's not me, however. Then there are the gap junctions. The gap junctions attach the membranes of neighboring cells, but form structures, the connexons, that allow the passage of components from one cell, from the cytoplasm of one cell to the cytoplasm of the other cells. The connexins are the main components of these gap junctions. And then there are the hemidesmosomes that attach the basal part of the epithelial cell to the basal lamina. And this attachment is done through the integrins. And the hemidesmosomes also connect with the desmosomes via the intermediate filaments, which we know in epithelial cells are the cytokeratins. The electron micrographs here, transmission electron micrographs, show here the hemidesmosomes, and here the junctional complexes, the tight junction. So there is no space really in between the membranes of the two attached cells. These are the attachment junctions, these are the zonula adherents, and these are the macula adherents, the spot desmosomes. These are the gap junctions. A nice drawing. This is a freeze fracture micrograph that shows you that the gap junctions contains all these channels, the connexons, through which they are the only ones that connect the cytoplasm of the neighboring cells. A few words about the specializations at the apical domain. I mentioned the microphyllae that are penetrations of the plasma membrane, the apical plasma membrane, into a lumen. The lumen would be the interior of the small intestine, and they contain acting as a cytoskeletal structure, and their role is to increase the absorptive surface. The other specialization that we discussed today are the cilia. The cilia are present at the apical part of the columnar epithelia in the respiratory pseudostratified columnar epithelium, that they have the microtubules as a cytoskeleton, and their property is to beat, and they beat in usually in one direction and generate a flow of fluid at the surface of the cells towards one direction. To recapitulate, features of epithelia are that they are avascular, they can transform from one epithelial cell type to another in the process of metaplasia. They can easily regenerate, so the wounds are repaired easily, and they can undergo not only metaplasia but also dysplasia. Dysplasia is a malignant change, and it involves not only a change in the shape of the cells, it also involves a genetic mutation. Once there is a wound in this epithelia, the cells reproduce very fast and seal the epithelial lining. Clinical points: the epithelial cells have high mitotic index and are exposed to the surface, which gives pathogens and carcinogens free access to them. Malignant neoplasmas of surface epithelium are the carcinomas, and those originating from glandular epithelium are the adenocarcinomas. the exposure to asbestos causes the pleural mesothelioma that I mentioned already. The transitional cell carcinoma is typical from the urinary tract and originates in the transitional epithelium. And the epithelial dysplasia is a premalignant change in epithelium, alteration in the cell structure encoded by the genome plus an abnormal appearance of the tissue. And this ends the second lecture, but unfortunately, I still have to go through a very small presentation of the virtual microscope lab. I stop this recording.